Okay, so let's start the proceedings. Uh, Welcome all to this very special webinar. I am Sudha Bhattacharya. I am co-founder of World Without GAD Myopathy, an organization that we set up with the view to spread awareness of this very rare disease in India and to have a patient cohort whom we can then help to get diagnosed correctly and uh, also in the absence of any treatment to help each other on how to manage the disease. Now, over the years, we have now started focusing our attention more closely on trying to develop and promote therapies for GNU myopathy. And that is the genesis of today's webinar. So GNU myopathy being a very rare disease, there is not too much research going on internationally and within India. But Duchenne muscular dystrophy is the disease which is much more actively being worked on. Uh, but there are a lot of common factors between the two diseases. Of course, the organ affected uh, being muscle and also other pathways. So therefore, we have a lot to learn from DMD. And today, Sonia will be talking to us about the technology that she has developed, Myo-Organized, that is how to look at a three-dimensional muscle tissue in the lab and to be able to use it to understand the disease and also to test new therapies. And Dr. David Israeli, he has done some really uh, cutting edge on the effect of lysosomal damage in DMD and the pathways that affect autophagy as a result. And the autophagic flux, which he is reporting in DMD, is also found in GNE myopathy. So we have some common factors there. And we are very excited that Sonia and David have agreed to talk to us. And we hope to learn a lot by listening to them. So I will now request Alok, Professor Alok Bhattacharya. He is the managing trustee and co-founder World Without GNE Myopathy. And he will do the formal introduction and take the proceeding forward. So thank you all.
Thank you, Sudha. It's my pleasure to introduce Dr. Sonia Albini. And before I introduce her, both our speakers are from this institute, Genethon. And long back, I had a privilege of visiting this. I think roughly about 7, 8 years back or so. And also the Institute of Myology in Paris. So I think Paris has probably one of the best centers for concentrated research on muscles, and this institute is playing a big role, particularly not only studying these muscle diseases, but also coming out with therapy, developing new technology, etc. Dr. Albini is a group leader in a Genethon, where her research unit is essentially integrating basic science along with translation. And she has more than 15 years of experience in muscle research. And particularly, she has a lot of expertise in iPSCs. And then, of course, two-dimensional, three-dimensional organization of iPSC. And her interests are a variety of progressive muscle diseases. And obviously, there is a major problem of generating, cellular or animal system that mimics the disease, because those are very important for drug discovery. And she has done tremendous amount of work in this. She holds a degree in PhD genetics and cell biology with a PhD. And of course, she has a master's in biotechnology. She has published extensively. So we're going to look forward to listening to you, Dr. Sai. So it's over to you.
Yes. Thank you very much for your kind introduction. I'm very happy to be here. Thanks a lot for this invitation. So yes, as you mentioned, so I am from Italy originally, and during my education, I spent many years in United States, always focusing on Duchenne muscular dystrophy, and it's there that I acquired expertise in the stem cell, embryonic stem cell system, and where I learned more about the disease modeling. So when I joined Genethon, I tried to combine this expertise with the opportunity to implement that for translational purposes, given the focus on gene therapy that we have at Genethon. So I will describe today the work on generation of iPS, patient-derived muscle organoids, a platform that I called Myorganoids, and application for translational purposes. Right. Oops.
It's not changing.
Yeah, that's fine. Can you see and hear me well? All right.
Yeah.
So here there is just a transition slide for you to introduce the topic of organoids. I don't know if you are familiar or not, but I will summarize really the need nowadays to generate and to use these models. In fact, organoids are a transformative tool to study human development and for drug discovery. And why that? It's because of the physiological relevance that we can have from organoids. Because they recapitulate the three-dimensional architecture and the structure of the tissue, they can also reproduce the function of the tissue, which is really the most important parameter when we need to study the pathophysiology of a tissue. So we need to be able to reproduce its function, which is dependent on the structure. So the 3D interactions between the cells and between the cells and the matrix, they are crucial to establish this mature organization. And so today we have several applications that can be associated with organoids, disease modeling, drug discovery, high-throughput screening, cell and gene therapy, but most importantly, because we can derive those structures from patient cells, we can go towards a personalized medicine, meaning that we can generate organoids which recapitulates patient's disease because they will carry the disease, the genetic mutation of the patient. And now there is also the recently approved FDA guidelines, which really are really helpful to guide the researchers on how to use and validate the so-called new approach methodologies, the so-called NAMs for drug development. In fact, in those guidelines, which have been released in March, it really stated how important it is to have a standardized, robust, reproducible, patient-centered models that could mimic patient disease, improve therapeutic prediction, and accelerate drug discovery, and also, very importantly, reduce animal testing for ethical reasons, but also to have a more predictive therapeutic efficiency, therapeutic prediction. So here, just a slide where I show why we use the induced pluripotent stem cells and what's the advantage of using iPS. So iPS are induced pluripotent stem cells derived from patient biopsies. And nowadays, it's very easy to get access to, for example, skin biopsies or urine samples from which, or blood samples from which we can derive PBMC and generate iPS. So those iPS have two major features. which make them very appealing for the research, that are the unlimited proliferation ability and the pluripotency, meaning that they can generate any kind of cell types of the human body. So the crucial challenge for the scientists is to generate efficiently specific cell types of interest, like in our case, muscle cells, for example. So in my group, we focus on 3 different axes: a technological part with the tissue engineering, and we focus mostly on muscle force assay as a readouts, the disease modeling starting from patient cells. As I told you, my focus is DMD, but we are also implementing this system to other muscular dystrophies like limb-girdle, for example, from tendinopathies. This is our second interest. And then we use this system to develop and test the therapeutic strategies. So we have both fundamental research program where we really want to investigate what happens at molecular level and identify targets, and the translational one when we use this platform to screen for our products or potential promising products. So I don't think I need to introduce a lot to Duchenne muscular dystrophy. You are familiar with this disease. I want to just remind you that the severity of this disease, which is still incurable, it's a pediatric disorder which leads to premature death for respiratory and cardiac failure, is caused by the absence of dystrophin. So there are several mutations of these super large genes, which is dystrophin, that leads to lack of proteins. And this lack of proteins affect the membrane stability. So the membrane is very fragile and undergoes repeated cycle of degeneration and regeneration, which ultimately leads to secondary pathogenic events such as inflammation or fibrosis. And this is very important to take in mind, because in those patients, so even when in very severe stages, where fibrosis is really prominent, gene therapy efficiency is really affected because of this fibrosis. So it's a really multifactorial disease at the end, besides the genetic mutation. So the most promising approach is the gene therapy, where we deliver the therapeutic genes by AV vector, adeno-associated virus, which have this capacity to target the muscle very efficiently. However, in the clinics, now we have 5 different products, which are all based on miniaturized version of dystrophin, because AV can accommodate only 5 kb of the gene, and the dystrophin is more than 11 kb. So it has been shown in animal models that those short forms are still functional, and indeed in the clinics has been shown to ameliorate the patient phenotype. However, the therapeutic efficacy is still partial. And one of the reasons, so one of the different challenges that are associated, in fact, that we learn from clinical trials are due to, we can have different explanations for the limited efficacy. One could be due to the fact that the short form of dystrophin lacks several key domains, but also to the fact that we don't correct secondary downstream events, and also that animal models, they're not maybe so predictive because of the species difference. So there is a limited translatability, and that's why there is really the need for a human relevant model to improve the translatability and accelerate drug discovery. So we can fill this gap of species difference by using IPS from patients, as I told you, that can be differentiated into muscle cells, for example, but in particular, we also employ biomaterials and bioengineering approach to derive 3-dimensional muscles. So we add a 3D scaffold matrix growth factor to replicate tissue structure and function, so that we can have a bridge between the simple 2D models and the animal models by using the 3D organoids. However, we have to take in consideration one important parameter when we want to embark into the organoid generation, because now you will know that there are several complexity of organoids. We can go from simple organoid to organoid chip. So there are several levels of complexity, but the higher the complexity, as you see in this graph, the lower the resolution and the throughput. So it's really important to compromise between complexity and throughput, and so really tailor the composition of our organoids based on the question we have, because too much complexity can introduce variability and the results can be difficult to interpret. So this is something to take in mind. However, organoid, as you can see here, it's a compromise between the throughput and the relevance, because they can replicate the tissue architecture and function. So in the work that was performed by a talented PhD student in my lab, is to development of a more exacerbated DMD organoids that we call profibrotic organoids for therapeutic screening, because the aim was to reproduce these fibrotic hallmarks, which is very important for Duchenne. So there are several protocols nowadays to generate myotubes from IPS, transgene free or transgene based. As you can see here in my US postdoc, I generated a protocol which is based on reprogramming, epigenetic reprogramming of the cells, transgene based, that I'm using still now with the students, and it is based on the electroporation of IPS with two transgenes. One is MyoD, which is a master transcription factor, and the other one is a chromatin modifier. And this protocol has an advantage to be very fast and to give rise to homogeneous population, because we bypass the formation of mesodermal progenitor. So it's really direct. We don't go through the long protocol that mimics the developmental cues. So it's really fast. In 7 days, we get myotubes. Here is just to show you an optimization that we have done to generate myotubes in 2D by just switching to commercial medium with our protocol. You see, by using those combination of those commercial mediums in this transgene-based protocol, we have 100% of myotubes. And here it's the differentiation potential of control versus DMD cell lines in 2D. Those are cell lines that we have in the lab, and I also have a pair here, isogenic cell lines. So one is mutated, the other one is corrected by CRISPR-Cas9. And so I will be showing this isogenic because there we don't have the variability of the genetic background. So the generation of the organoids follow this protocol, which is anyway published, but you can see here that we do a casting of committed IPS and fibroblast. We keep it in 2 days in growth medium and other 12 days in differentiation medium. And then we have the formation of a ring. The ring is formed because we used a 48 well plate from Myriamed that are equipped with the 2 silicon pillars, flexible. So this allows the growth of the tissue in a ring shaped format. And here you can see, and just wanted to emphasize the fact that we added fibroblasts, because fibroblasts, they are a very important component of the connective tissue, and they really help muscle assembly. So we tested several ratio of fibroblasts, and we found that 10% of fibroblasts was the best concentration, was the best ratio for us to achieve alignment of the myotubes as compared to the other conditions. And this also mirrors what is happening in the normal muscle, where we have around 5-10% of stromal population. Here is the kinetics that show you the gradual compaction of our organoids from day 1 to day 14. The inclusion of fibroblasts makes the compaction more efficient and also faster. Most importantly, the inclusion of fibroblasts we show that allows higher structural organization maturation. You can see the higher maturation of the architecture, because the fibroblasts, including my organoids, are more aligned. The alignment is calculated by the angle between myotubes, and the fiber are very circular, as shown by the feeders diameters XY ratio. And in line with that, we also observed an increased maturation. Here in the black and white, you can see, in fact, the higher width of the Z disc. So it's electron microscopy. They show you the more clear formation of Z disc, and also the higher level of striation and multinucleation. And finally, in red here, you can see the staining for dystrophin, the membrane marker. As expected in mature muscle, dystrophin increases, and also its localization on the membrane. And also, we observed an increase of the late muscle gene markers like myosin heavy chain 2 and 7. And so, in line with that, we evaluated the muscle function by using a really sophisticated, let's say, organ bath system, which allows the measurement of muscle force from these organoids. So this is really a direct force measurement, so where we use electrical stimulation in this muscle strip chamber. And as you can see here, the ring are placed around the hooks and can be stretched. This is one strength of the system. It allows the stretching of the organoids. And this is important in muscular dystrophy because it allows to perform eccentric contractions, which are very important to assess fatigability and muscle resistance. So here you see that we first optimize the force-frequency relationship, and so we identified this frequency as the best, showing the highest peak. And we observe the increase of tetanic force when fibroblasts are included. So higher maturation, higher function for the muscle. And so from this point, we decided to use fibroblasts also derived from DMD patients to try to exacerbate the disease, with the idea to increase, to mimic the pathogenic environment within the muscle. And this is why, and this is because fibroblasts can sense. There is a crosstalk between muscle and fibroblast, because they can signal each other, and the fibroblasts tend to secrete more matrix because they are activated by the damaged muscles. So it's really important to mimic this crosstalk. So we use as fibroblast lines human immortalized control NDMD lines that we get from Institut de Myologie here in Paris. And we cast those fibroblasts with IPS, both from control and DMD. Sorry, there was a noise. And we generate organoids, and then we perform muscle force protocol to assess fatigability. The eccentric protocol consists in giving 10 eccentric contraction repetition, where the muscle are stretched, are length, And then we calculate the difference between the last isometric contraction and the very first one before the eccentric exercise. And from this, we can have the force loss by this formula. And then we also look at the half decrease in time, which is also something that is impaired in Duchenne. And also we demonstrated that with this assay, with this eccentric control repetition, we are able to recapitulate the membrane damage because if you see the yellow, when DMD include fibroblast, basically they are positive for staining with Evans blue dye, which is a marker that gets incorporated when the membrane is permeable. So it's a damage marker, and you can see very well that in the DMD this marker is higher. Notexin is just an injury control. And so here you see that when we include fibroblasts in the organoids, if you just see the difference between the control, the blue line, and the orange line, there is a separation of the group. So there is a statistically significant difference between the groups. And so the force loss and also the relaxation time. So basically, the inclusion of fibroblasts exacerbates the disease phenotype and allows discrimination of this hallmark of muscle dysfunction as compared to the control group. And then we looked at the transcriptomic signature after this exercise protocol. And we could observe that there is a profibrotic signature here in red in the mutated cell lines, which is very visible, and also activation of the TGF-beta pathway in all DMD lines. TGF-beta pathway is visualized by accumulation of phosphosmad in the nucleus, also visible in the staining of organoid sections. You have an accumulation of phosphosmad in both myotubes and fibroblasts, meaning that we can recapitulate the known activation of TGF-beta signaling, and most importantly, a profibrotic signature in those organoids. Here is just some validation where we see a relative gene expression of fibrotic genes that are highly upregulated in DMD organoids, including fibroblasts. And so far, those data highlight the fact that with this system, we could reproduce two key hallmarks of Duchenne: muscle dysfunction and profibrotic signature, and also establish, therefore, two important readouts that we can further use for screening. And in fact, as a proof of concept, we use AV gene transfer of microdystrophin to see if we could mimic the improvement that we see in the clinics. Here is just a reporter system where we can see the efficient infection of the muscle organoids with AV. This is just a reporter system. You can see that AV can be delivered very well in the organoids. And here we use different titer to identify a low versus high dose, for example. And here we have data with the microdystrophin gene transfer. So basically, you can see that microdystrophin can be delivered efficiently, as you can see by the calculation of viral copy number of microdystrophin mRNA expression and also protein level in a dose-dependent manner. So we first look at this transfer efficiency, and then we looked at the muscle force analysis following microdystrophy. So as you can see, the protocol can basically show that there is a dose-dependent amelioration of the half decreases time and also fatigue index for all cell lines. Only in this last one, basically there is not significant, but there is a trend. So basically there is a great improvement of muscle force loss. You see, in fact, the reduction of the loss using high dose of microdystrophin. However, it's not a complete recovery. And here you can see histologically the presence of dystrophin in the muscle sections. Dystrophin is in red, and here just to show you that dystrophin basically it links the cytoskeleton with extracellular matrix, and so it's a bridge that stabilizes the membrane. So when we want to see the efficacy of dystrophin, besides looking at dystrophin localization, we need to see whether the dystroglycan complex is restored. That's why we look not only at microdystrophin, but we also looked at those two crucial components, which are alpha and beta dystroglycan, to see the reestablishment of the dystroglycan complex, of the link. And you can see here that although we have in the optimal condition, in the high dose, a total restoration of dystrophin, the restoration of alpha and beta dystroglycan is not complete, especially for alpha dystroglycan. And so this was a kind of unexpected, because we see an amelioration of the phenotype, we see an amelioration of the muscle force, but the complex is still not completely restored. And at the same time, we also saw a partial rescue of transcriptomic signature. In fact, here it's the profibrotic signature in the control, here on the right in the mutated cell lines, and look here in the middle is the DMD with microdystrophin. You can see that most of the genes are still dysregulated, meaning that fibrosis is still persistent. And here, in fact, you can also see by the gene ontology enrichment analysis that there are pathways that are corrected by microdystrophin, but many others that are not corrected by microdystrophin. And here, for example, you can appreciate TGF-beta signaling and many extracellular matrix proteins or collagens, tissue remodeling enzymes, which are still very up despite the improvement in muscle force by dystrophin. So basically, this model shows that we can replicate hallmarks of DMD, and we can therefore use that for gene therapy screening, with the potential to unravel limitation of current approaches, for example, besides serving as a screening platform. Here is just some applications that we do in the lab. We use this like a screening platform to test, for example, AV-mediated strategies. So not only gene therapy, but also, for example, lentiviral LNPs strategies or pharmacological approaches. So this system can be suitable also for other kind of delivery system, and using readouts of force analysis, transcriptomic, and secretome. And one of the projects that are going now in my group is the inclusion in the organoids of fibrodecompogenic progenitors, the so-called FAPs, which are the muscle fibroblasts. It's a very important population which plays a major role in the accumulation of extracellular matrix. So during disease progression, these FAPs, as you can see here, switch from a healthy, from the progenitor state to a pathogenic state where they differentiate into myofibroblasts or adipocytes. And so they basically are the major source of fibrosis in the dystrophic muscle. And so we want to understand how the dysregulation in cellular communication can bring to these pathogenic states, and for that we will perform a single nuclear RNA-seq. And So basically, we act on different levels, the cell complexity introducing FAPs and the microenvironmental control, because we can modulate the response of the cells by extracellular cues, for example, by fibrotic or inflammatory cocktails. And of course, for long-term studies, other systems, including microfluidics, could be beneficial because one limitation of this system is that cannot be kept for very, very long. So for long-term studies, of course, more state-of-the-art technologies are also needed. So here is just acknowledgement of the people and the grants, the funding, and Genethon facility. So I will be very happy to take any questions if you have them. Thank you so much.
Thank you. Thank you very much for a wonderful talk. I mean, we learned so much from it. I think we'll take up the questions in the end. Is that okay?
Okay. Yeah, sure.
Okay. So, please, if you have questions, you can put it in the chat box and we'll put it up with the speakers in the end of the talk. Thank you. I move on to our second speaker, Dr. David Israeli, and he's going to talk about overcoming lysosomal insufficiency and for improved gene therapy in DMD. Dr. Israeli received his PhD in cancer genetics from Weizmann Institute of Science. Then he was awarded Marie Curie Fellowship, postdoctoral grant, and he has been with Genethon system for quite some time. And he's currently has a research position again in the progressive muscular dystrophy group of Genethon. And he's essentially working on DMD with trying to figure out all the various defects that it could be. And between them, I think they're going to sort out this problem of DMD and eventually come out with a biomarker or a measurable marker for testing any gene therapy or any other drugs for that. It's a very wonderful to see that there are systems that can be actually used to get a more human-like phenotype that can be assessed. Dr. Israeli, go ahead.
Yes, thank you very much. Can you see my screen now? I don't know.
Not yet.
Not yet, because I try to share it. I'm not sure I'm doing it correctly. Is it okay now?
Yeah. It's not fully coming. Yeah.
It is.
That's fine.
Okay, great. So yes, so thank you for the invitation, the kind introduction. And I think let's go directly into the business. So I don't need much Duchenne introduction after Sonia that gave you some minimum, assuming that you know what Duchenne is. And so it's just kind of generic introduction slide into Duchenne, which is a terrible disease that although it's a rare disease, it touched quite many people worldwide, and you know it is evolutive and it is ended almost inevitably by the death of the patient from cardiorespiratory failure. And so here I'll just give you the bottom line, and then the bottom line is of this seminar is this paper that you see here that we just published. And so I will not go into details, but the paper, of course, is available, and you can go into details if anything is not entirely clear. But the message is here, the take home of this paper and of this seminar, of which I'll coming at the end too. So we found that lysosomal damage is a key feature of DMD. This is quite unknown. And also what we have found is that gene therapy alone cannot completely restore this lysosomal function. Because you know that a gene therapy in Duchenne is imperfect, probably because we're using the microdystrophin and not the entire dystrophin. And also what we show that if we can target lysosome in combination with gene therapy, we can get improved results. So historically, now I do zoom out and I'll explain you how we got over the year into this story of the lysosome. Because if you go now and you look for in the scientific literature for Duchenne and lysosome, you don't find very few, in fact. So, but historically there was some papers over lysosomal function. And so you can see the 80s, the 70s. One of them came from Viktor Dubovitz, who's the founder of the World Muscle Society and the Journal of Neuromuscular Disorder. And so, but normally it was used to be as something which is associated to the disease. It was, for instance, used for detection for analytical purposes. It was not thought to be a major driver of the disease. And globally speaking, until the late 80s and the beginning of the 90s of the 20th century, Duchenne was thought to be a metabolic disease. And as you can see here in the left part, here's a medical hypothesis. But you must know that the discovery of the dystrophin gene at the late 80s, beginning of the 90s, completely changed this vision, because once that gene was discovered, sequenced, and function analysis, structure function analysis provided its spatial localization as a linker of the inside and the outside of the myofiber, it was intuitively clear that dystrophin functioned mainly as a structural stabilizer. And then for the entire way of understanding of the disease shifted into more, let's say, structural mechanical vision and not that much metabolic understanding of the disease, which is something that was a little bit abandoned among researchers. And now let me do a zoom out into how we came into this lysosomal, and the story is begin quite many years ago, and it began for me with the first clinical trial that was run by Genethon. This is almost 20 years ago, and so that was not in Duchenne, that was in limb-girdle muscular dystrophy, gamma sarcoglycanopathy, and that was relatively simple intramuscular injection of the transgene, not injection, AAV, transduction intramuscular of the transgene. And as first experience of Genethon in muscle disease, in gene therapy in muscle disease, the very few things that were confirmed by this in this essay was that transgene was correctly expressed. There were no safety issue, and AAV production appeared to be a problem because you need huge amount, and also they noticed some immune response. So, and the point is that also one of the things is it has been it was understood that biomarkers for evaluation are so much necessary, because when you treat in gene therapy, in this type of disease, you don't expect very quick functional improvement. So you need to develop biomarkers that will tell you if it works or if something is working before a real therapeutic benefit can be observed. So we have embarked into a project of biomarkers. And we and other group at more than 10 years ago published that circulating microRNA can be used as biomarker. And in particular, in the beginning, we have identified a group of microRNA, which is called myomere, because they are expressed enrichedly in the muscle. And so these miRNA happen to be very much upregulated in the circulation of mouse models. And so it has been concluded that yes, the technology of looking for biomarker with circulating miRNA might be interested. And so we moved into human large cohort of human DMD patient, and we just did the same thing in order to see if we can recapitulate these result. And so what I'm describing you here, so there was a large cohort, DMD patient, 81 people. And so we run the same study for looking for perturbation of circulating miRNA in DMD patient. And here we had surprise. And the surprise that we had that what I told you before, in animal experimentation, we just found a small number of microRNA which were dysregulated in Duchenne. And when we moved to a large human cohort and also improved technology of miRNA, what we have found is tremendous level of perturbation of circulating miRNA in these patients, Duchenne patient, and we were quite surprised. And with these results, we in fact we ask ourselves, what's the reason for this huge level of perturbation of microRNA in Duchenne muscular dystrophy? And the answer, there was no answer. The answer is that no one knows. And so we said it gave us indications that we still very badly understand this disease, because none of the things that are known of Duchenne muscular dystrophy could explain that. So we said, yes, biomarker is an interesting field, and we have now many biomarker candidates in Duchenne muscular dystrophy, but perhaps we could do more than biomarker. Perhaps if we understand better the reason for this perturbation, we can identify new things about the pathophysiology of Duchenne muscular dystrophy. And here we moved into a classical problem of omics. In omics, very often investigation of omics, you end with a lot of perturbation, and then you don't know what to do with these huge amount of data, how to develop analytical system or bioinformatic capacity to analyze in order to understand and interpret. And when it comes to miRNA, so this is, I don't know if you see my flash. Can you see my flash or not? Can you hear me? Or I'm talking to myself?
Yes, we can.
Can you see my can you see my my flash on the screen or not?
Yes.
You can see. Okay, here it is. So normally, so this is the red guy is microRNA. And in this case, a microRNA is located internally in intron of a host gene. And this is true for about half of the miRNA. They reside within host genes, while other miRNAs will not be, they will just be somewhere in the genome, not inserted into host genes. Now, classically, what people do when they try to interpret miRNA results, they will ask, you know, that miRNA target, miRNA are regulating expression of the target genes, and each miRNA may have up to a few dozens or up to 100 of target genes. So people that try to understand miRNA, they're asking, in fact, what the target genes are doing. So the problem is that you have so many of them, and it's not sure if all these are regulated in your relevant tissue. And so it gives rise to prediction which are not very much reliable. And we said, instead of interested in all these target gene, what happens if we get interested in the miRNA host gene rather than target gene? And the question is, in fact, is phrased here. We stopped asking what miRNA do, and we start asking why they are dysregulated. And the answer is these miRNA are dysregulated very often because the expression is associated to the expression of their host gene. So What's going on? I cannot move forward now. Why? Excuse me, I have technical issue here.
You have to hit escape and then you'll be able to move.
Yeah, but non-escape is not responding. Oh, okay. Here it is. So I just give you a quick example of the principles of the spatial relation between intronic miRNA or exonic, because they can be also in and their host genes. And this coming from the work of Eric Olson, very well known in muscular dystrophy. So in 2009, what they have shown that a group of miRNA, which are known as dystromir, because they are also associated to muscular dystrophy, they are host inside the myosin heavy chain. All of them are important, of course, for skeletal and cardiac muscles. And what the paper said and show is that this co-regulation by the miRNA and the myosin heavy chain, the sulfate and the myotube type. And so this is just demonstration that if you want to understand best the biological function of these miRNA, you're not going to ask about the target genes. In fact, you can learn from the host gene, which is a myosin heavy chain. And so this is just demonstration why host genes can better explain sometime the biology as compared to the target gene. So so we had, in fact, in our screener system, we had many miRNA dysregulated, and I just try again here to, because we cannot see my titles in, so I try to arrange it so that you can see the titles of my. How do I get rid of this? Well, I don't know. It's stuck there. Sorry for this. I don't know how to get rid of it. Okay, let's leave it like this. The problem is that it hiding the title, so looking if we can.
So you're talking about that moving control, right, in the zoom?
Yeah.
So there is a three dots there on the top right hand side,
Yeah.
and then you just hide the floating thing.
Yeah, but I don't have this.
I see.
Sorry for that. I won't be able to do that.
Or you can just drag it down.
Yeah. Yeah, the problem is you're so huge that it is kind of how do you deactivate all of these problems associated. We use more Teams and Zoom and so maybe. Okay, listen, I'll continue this way because we're losing time and I don't know how to get rid of that unless I reinitiate the whole thing, and I don't want to do. So yeah, so it's hiding my title. I tell you the title. So go back to the story of host gene. We had a huge amount of dysregulation, and so we could find 71 host genes. And so now, if we just took these miRNA and we do target gene prediction, what we got is normal things that you see in muscular dystrophy. We got a prediction that ECM receptor interaction are perturbed in Duchenne's, focal adhesion, PI3, AKT signaling, and FOXO signaling. In contrast, when we looked what happened with what will be the prediction of the host gene, we had the surprise that the prediction, the bioinformatic prediction, was a perturbation of lipid metabolism, and in particular, cholesterol are associated to Duchenne muscular dystrophy cholesterol perturbation, and that was completely new. Of course, we didn't expect, and we didn't even believe. Not only we didn't expect, but we decided to stick to it, to look at the literature, and so I'm sorry, I'm having. I think I have to quit and.
But we can see the title, eh?
Maybe I have to. I lost myself now. This Zoom is giving me problems. Can you see now my screen?
Yeah.
Can you see my screen?
Yeah.
Yes.
Okay, so let's
Okay.
continue. I'm sorry. When thinking about cholesterol perturbation, we often intuitively think about circulating, of course, circulation. But we said maybe it's not the circulation cholesterol because we're in a muscle disease. So what happened in the muscle? And at the same time, there was a paper that was published in the United States in a PNAS paper that was showing that if you treat mouse model, the MDX mouse model with simvastatin for the removal of cholesterol, you get improvement. They were thought this is related to circulating cholesterol, but we thought it might be related to the cholesterol in the muscle. So what we have done, we treated the MDX mouse with simvastatin, and indeed what we noticed that in the MDX mouse, you get increased muscle cholesterol, and when it comes after treatment with simvastatin, you get reduction of cholesterol. And also what was interesting that this in simvastatin treatment, it reduced, and in correlation, it reduced some of the parameters of the disease. It improved some parameters, not all of them, but some of them. And so from that, we said, so we have too much cholesterol in the muscles. And so what's the sense behind it? So what does it mean that you have too much cholesterol in the muscle? And because there's so few data about the cholesterol in the muscle itself and possible effect. So we went to see what known about perturbation of cholesterol in other disease. And this in particular was studied in the central nervous system and in different model disease and in patient like such as Niemann-Pick and Alzheimer and Parkinson and Huntington. In all these diseases, was reporting in the scientific literature that if you have too much cholesterol, it will be associated to lysosome perturbation. And we said maybe we have issue of lysosome also in the muscle. And so where are we with that? We hypothesize that cholesterol alternation reflect lysosomal damage in DMD muscle. So muscle cholesterol was not linked before to lysosomal impairment. However, as I told you, in neurological, it was a case. And so we found in the literature that galectin-3 is a biomarker for lysosomal damage. And then we looked at the databases of gene expression in Duchenne patient, and we found that galectin is tremendously upregulation transcriptionally in transcriptomic study that came from animal models and also from patient. So we said, okay, if this biomarker of lysosomal damage is so much upregulated, yes, maybe we do have issue of lysosome in the muscle. So let's use precisely this biomarker in order to see if we can get if there are damage of the lysosome in the muscle. And so this is what we do here. So normally the lysosome is a double membrane encapsulated, and once you and galectin cannot go in, but once you have damage of the membrane of the lysosome, what happens that glycans in the lumen side of the lysosome are exposed, and the galectin will go in and will make what's now termed galectin-3 puncta. And so to test whether this is happening in the muscle, these are myoblasts and these are myotubes, and they are from normal. They are not Duchenne, but they are treated with a poison that LOM it's called. It's a chemical that will make induce lysosomal damage. And what you can see here is that either on both in myoblasts and in myotube, when they're treated with this damaging agent, what you can see here is these small dots. These are the puncta assay. These are the puncta that mark lysosomal damage. And so then we move to look what will be happening in the mouse. So what you can see here is that in the muscles from, this is a gastrocnemius from the wild type mouse and from the Duchenne mouse. And of course, you can see tremendous upregulation of galectin. And more important, now we look at transversal sections from the muscle of respectively the wild type and the DMD mouse. And what you can see is overlap between LAMP2, which is a lysosomal marker, and the galectin-3. And clearly, the lysosome inside the myofibers are fully showing these signs of lysosomal damage. And here on the right, you can see the puncta, which is tremendously upregulated in this mouse model. And so that was indeed indicated that there's ongoing lysosomal damage in the muscle in this mouse. And so here is some more characterization of this phenotype. And so what you can see is that LAMP2, which is a marker of the lysosome, it's upregulated in the MDX in the Duchenne mouse. And also, what you can see is that this is size distribution of the lysosome. So you can see perturbation in the size and of the lysosome. And also in the numbers as illustrated, we have a larger number of lysosome, and they are showing all kind of strange, all kind of perturbation. And so here, what you can see, so here is a nice illustration which was made from single fiber. So these single fiber, so reconstruction in confocal, and what you can see when they are stained also again for the galectin, for the damage of the lysosome, and for LAMP2. So what you can see is that in the Duchenne mouse you have much increased number of spot and increased size of the spot as illustrated on the red on the Duchenne mouse. I just skip it. And also what you can see here is that, so this is electron microscopy of the MDX mouse. And again, when you compare to the wild type lysosome, muscle lysosome, to the one of the MDX lysosome, what you can see is that the lysosome are bigger and filled with stuff which cannot be degraded by the lysosome. And so, of course, this is also a functional problem of the lysosome, because when they are clogged with all these materials, there's strong dysfunction of the lysosome. So this dysfunction of the lysosome, it's not only functional, but morphological, can be seen morphologically. And then interestingly, this is not limited to the MDX mouse, because we also found in a dog model just the same. So here you can see the puncta, the galectin-3 puncta, which is visible in the skeletal muscle of the dog model, and also in DMD boys. So just the same increased number of puncta in the muscle of healthy patient, of healthy person. In fact, there's no any puncta visible by this technology, and you do see huge amount of lysosomal damage in the patients. And so, so now let's shift quickly into gene therapy. And in gene therapy, gene replacement, what we do now, also in human patient, the microdystrophin, it is increasingly used also now, approved by the FDA, and we're using in Genethon exactly the same construct in animal experimentation. And here what you can see is dystrophin is visible in a wild type, completely absent in the mouse model, but then when you transduce it with the AAV, with a microdystrophin, increasingly in a dose-dependent manner, the dystrophin is come back, and the fibrosis is gone. As you can see, dose response increases of dystrophin, global force, myomesine is a marker of damage, like the creatine kinase, so it goes down, and fibrosis also go down. So this just to illustrate what gene therapy does in this mouse model and also in patient now. Also, you can see also in global force, which is improved with the treatment, and it's just the same illustration that after treatment, so it goes down. Sorry, yeah, because of this is hidden here. This is not the Duchenne. This is okay. Now you can see it. I'm sorry, it's smaller, but at least it's visible. So here is another. Here we move into gene therapy of another muscular dystrophy because, okay, I'll go back. One of the things that you can see here is that the gene therapy, yes, it is working, but it's not perfect. Why it is not perfect? Because even at a higher dose that you have 100% of the fibers expressing dystrophin, you don't get back to normal in terms of global force is still below normal. In terms of marker of damage like the myomesine, you don't get back to normal. And also in terms of fibrosis, there's still different between the wild type and between the treated mouse. So you don't get completely back to normal. Now, what we have done that we move into another model. In this model, we're talking about LGMD-R5. LGMD-R5 are gamma sarcoglycanopathy. This is disease with phenotype quite similar to Duchenne muscular dystrophy because it's the same dystrophin complex which is disrupted here. And the story is just the same. You do gene therapy with AAV. The thing is that gamma sarcoglycan is smaller. So you put the entire gamma sarcoglycan in the AAV and then you do gene therapy. And when you do that, you get almost complete back to normal. And so gene therapy in gamma sarcoglycanopathy in the mouse at least work much better than in the MDX mouse. And so this is important. And so what we do here, we compare the gene therapy in the Duchenne model to the gene therapy in the sarcoglycan model. And what you can see that for instance, in particular, the parameters of lysosomal damage, this galectin-3, is not coming back at all to normal in treated mouse. These are treated, this is a wild type. Here you have the Duchenne, and here you have treatment at low dose and at high dose. Even high dose of microdystrophin cannot remove the lysosomal damage. In contrast, on the right side, when I go to gamma sarcoglycan, what you can see here that upon treatment, the lysosomal damage is almost went back to normal. So this suggests the persistence of lysosomal damage in the model for Duchenne muscular dystrophy even after the treatment. Which is bring us to the subject of combined therapy. So what is a combined therapy? In the combined therapy, you want on the left side, you treat by your virus, and on the right side, you look for complementary metabolic or other treatments that will complement, as simple as that. And so we previously, attempted to do combined therapy in the Duchenne model with simvastatin, I told you, and so we treated by microdystrophin plus simvastatin. We got some improvement in some parameter, we got less improvement in other parameters. All over, the conclusion was that simvastatin cannot complement correctly the microdystrophin. However, this is also illustration of about what it means combined therapy. You put the virus on the other hand, complementary treatment on the other hand, and you're looking for your results if it is improved as compared to gene therapy only. And so what to take? We looked for something that can boost, of course, lysosomal activity. And the choice was of a drug which is called trehalose. And I'm saying a drug, but it's not a drug. This is just a kind of a sucre. It's a very simple sucrose-like. And so why choosing it? Because it was shown to be able to boost lysosomal function. It is safe, it is easy because it's available orally, and also it's FDA approved in a number of indications. So so the setup here of the experiment in the mouse model was, again, you compare the wild type to the MDX, and then you treat with one drug, you treat with the other drug, or you treat with the two of them together. And what we had here is, and so what you can see is that we had here that for most parameters, microdystrophin was better than the trailers. But surprisingly, what you can see is that the combined, first of all, was always better than the microdystrophin alone, as, for example, here in size distribution. So this is normalization of size distribution, which is abnormal in the MDX. It was normalized better with a combined. Fibrosis went a little bit down. What went tremendously down is a transcriptomic, because the transcriptomic signature, you see that this is the MDX, and the combine is here very close to the wild type as compared to the untreated or to the treated with only one of them, which remain far away. And also what's very interesting, that you can see that with trailers only, this is a trailers only, the lysosomal, so this is galectin-3 marker. This is shown here, I show you here. If you treat, this is the galectin damage, the puncta, galectin puncta, enough to treat with trailers only to remove these lysosomal damage. And of course, in the combined group, we get the best results as illustrated here. And another similar marker is IgG positive. IgG mark necrotic fibers. So you reduce tremendously only by trailers these necrotic fibers. And of course, the combined is best. What's interesting to for these parameters, which are lysosomal damage, the trailers is even better than the microdystrophin. And of course, together, it works best. This is almost the end. What the bottom line, the message of this presentation is that we possibly identify a new translation axis in Duchenne muscular dystrophy. We believe that DMD involves lysosomal dysfunction, something that was underappreciated or completely overlooked. We think that this is can be at today with the gene therapies that we have in Duchenne muscular dystrophy, which is partial with a microdystrophin, we don't get complete restoration of lysosomal damage. We showed that with lysosomal targeting strategy, you can improve the therapeutic outcome. And what we think that combined therapy is a promising approach for treat. And the last and the most important, the people who did that. So I told you, in fact, what I showed you is overview of almost 20 years of research. So the person who did most circulating microRNA work now more than 10 years ago, Fatima Amor, and then 2 PhD students in the group, Ai Vu Hong, which was much doing cholesterol perturbation, and Abbas Jaber has defined the lysosomal damage and the combined therapy. And now Ai Vu Hong and Abbas, the two of them are independent researchers in our group. And here the entire group and collaborator. And that's it for this presentation. And I'll be happy to take your questions. Anybody? Can you hear me? Hello?
Yeah, we can hear you.
Okay, can you hear me now?
Yeah. No, I was a little bit, because of all these technical perturbation, I was panicked. Maybe I'm talking to myself. So because
No.
without feedback, we
It
never
was a
sure.
wonderful thing, lot of food for thought, and lot of interesting aspects of various kinds of. I think we find some kind of link between different myopathies, etc. There was some question for Dr. Sonia, but she has already answered them in the chat box. So I need not go over it again. Do you want to, Dr. Sonia, you want to just summarize your answer to the questions that were raised? Sonia, can you hear me?
Sonia, you're on mute. I don't know if you're trying to speak, but
Yeah.
you are on mute.
Okay, I mean, I think most of it she has already answered it. There is a question from Sudha to David, and that was what are the other important host genes for 71 miRNAs? I think.
It's difficult to say. However, the table 70 of them, so many of them associated to lipid metabolism, of course, because so the bioinformatic analysis so this is gene network analysis of 71 host gene gave us in particular indication of lipid perturbation. So yes, this is the most things that are coming. Two of them, yeah, there are two particularly important is SREB. SREB is SREB1 and SREB2 is two master transcription factors that controlling SREB1 control fatty acid and metabolism and biosynthesis, and SREB2 control cholesterol biosynthesis. SREB1 and SREB2 have been discovered by two American scientists many years ago, and which got Nobel Prize for that. And very few information on SREBs in the muscle, but we have found that they are in Duchenne muscular dystrophy upregulated, and so the SREBs are controlling the cholesterol program and certainly involved in this perturbation. And because it's a transcription factor, also the targets are found to be dysregulated. So amongst them, among the target of SREB is enzyme which are controlling cholesterol, like the HMG C reductase, which is a target of the statins, and also is the SREB1 target are in fatty acid synthesis pathway.
Okay, so this is a question to both of you. This trehalose and your cholesterol thing work in the myorganide model. I mean, what is the result that you get if you try it out?
Yeah, I answered partially. Yeah, I answered now in the chat. We have done a preliminary experiment just with trehalose, and there was a slight increase in the muscle force. So we just talked about David. Now we are resuming the project. We will try to redo this in combination with gene therapy, though, because the idea is that they act in synergy. So we're trying the combinatorial strategy of trehalose plus microdystrophin in organoids. So, but the very preliminary data, there was this slight increase in muscle force, so it's promising, but we need to repeat. And we also tried to add cholesterol in organoids to mimic this cholesterol accumulation that we have in organoids, and also we had this slight reduction in muscle force, so it seems that the cholesterol is doing something. We are still working on recapitulating this cholesterol accumulation in the myorganoid model. So we are just preliminary data. We are working on it.
Okay. So this is a question from Rajesh to David. He's asking whether you've tried rapamycin alongside microdystrophin gene therapy, and also have you tried mini dystrophin?
People have tried rapamycin in Duchenne muscular dystrophy. I'm not sure if that was together with microdystrophin or alone. I think results were kind of mixed, and the thing is rapamycin and global M2C inhibition. This is a good approach theoretically because the M2C is hyperactivated, and one of the things that trailers do, and so the AMP kinase, it's upregulated the AMP kinase pathway to restrain the hyper M2C. So the rationale is there, but in reality, if you completely block M2C with rapamycin, you get many complications like it touches the immune system. And so I don't know if anybody tried. We did not try. Whether other people combine with microdystrophin, I don't think so. What happened when people put rapamycin only on the mouse model? Yes, it has been done, and I think results were kind of mixed.
Yeah, the other question was that I think there's a difference between micro and mini dystrophin, whether you see different results with mini dystrophin.
You want to take it, Sonia?
Both of you.
Yes. So yes, we have another project indeed where we compare different size of dystrophin, both in vivo models and in vitro model. So we have shown that adding key domains in dystrophin by using 2AV, because in one cannot fit, so we need to use a 2AV. There is an improvement, especially in the mechanic of the muscle. So you achieve a better membrane stability. But this is something that requires more sensitive readouts to be assessed, because in the MDX mouse, which has a mild phenotype, microdystrophin works already very, very well. In theory, it's enough. But in the clinics, there is a partial improvement. So the other effort is to understand whether adding several domains can improve the therapeutic benefits, and what readouts we can use to assess this in a sensitive way, to unravel differences. So we are setting up also different, let's say, muscle force specific assay, both in organoids model and in situ muscle model, animal model, always using, let's say, isotonic contractions, so meaning eccentric and concentric. So really mimicking the real world movement of the muscle, not the static one. So those are the two efforts we are doing now.
Okay, thank you. Swaminathan has a question that whether the Cellular treatment improved the longevity of microdystrophin. I guess he wants to see whether the effect survives a longer time if you give Cellular.
That's excellent, an excellent question, because before, if ever we reach clinical trial, we have to do long-term, we have to move to other preclinical models, and it's currently we will try to set it up, and we don't have the answer yet. What I can say, which is interesting and related to that when we checked lysosomal damage in the model, we do see lysosomal damage even at the age of 1 year old. It's not only initial, because what happened in this model that initially you have tremendous fiber breakdown and the disease in its peak at the first 2-3 months, and then the disease goes quite down with age, which also happen in human patients after the age of 10 or 12 years old. And despite the fact that the severity of the disease, of the symptoms is going a little bit down with age, with loss of fibers also, well, we do see in the mouse that even at old mice, lysosomal damage is persistent. But whether a long-term Trehalose treatment does the job, we don't know yet.
Okay. I think these are all you are in the process of doing many of these experiments. There's a question from Sudha for David. Are you trying any modulators of autophagic flux as well?
So one of the observations in the paper, which is developed more in the paper and did not show these details here, is that when you have lysosomal damage, your autophagic system is of course affected, because the autophagosome is a fusion between the phagosome and between the lysosome, or the autolysosome is a fusion between the autophagosome and the lysosome. And if the lysosome are not there, this fusion step, which is the last step in the autophagy, the last step is a lysosome. And so when you have lysosomal damage, what we have shown that also autophagy is affected. So we did not do autophagic flux experiment, but what we have shown that after the treatment of trehalose, we get improved parameters of autophagy. Yet I agree that we did not do exactly flux experiments. It will wait for another occasion.
Okay. So there's a question from Dr. Vishwanathan. And what do you think of combining anti-inflammatory agents with anti-fibrotic agents? I mean, he's a clinician, he's treating patients every day, so he would like to know that.
Yeah, we think that many of the things that were showing beneficial effect in preclinical experiment are working through lysosomal improvement. But there's one exception for that. Glucocorticoid, which of course are gold standard, does not seem to work through the lysosomal improvement of the lysosomal system. So I think this is not about fibrosis, but this is about the possibility to combine the glucocorticoid, which is anti-inflammatory and also metabolic regulator. I think it makes sense, let's say, to consider mixing of glucocorticoid with lysosomal boosting. Now, concerning anti-fibrotic, I'm not sure what drug exactly. The mechanism of fibrosis is so multifactorial and so complicated, and so I'm not sure if you can specify to which drug specifically.
Like perfenidone.
Maybe we can try to
Perfenidone.
figure out.
Perfenidone is there, right? There.
Yeah, I'm not sure. I have no answer on that.
So
I'm not sure. I must precise that I'm not a clinician, so when it comes to treat human patient, I'm certain things. Yeah, I miss certainly.
So Amrish has questions that sirolimus is being tested in clinical trials, pre-microdystrophin dystrophin infusion. And
what's the question? Sorry, I didn't catch it.
Anyway, Amrish, can you tell your question? Because you have two questions, you can combine them. Amrish, are you there? Anyway, I have a question before he comes on.
Okay, can you hear now? Yeah,
Yeah.
it was just related to the question that was asked about rapamycin, whether that can improve the efficacy of microdystrophin. So it was just related to that sirolimus is already being used in clinical trials. It was not a question, but just in context to rapamycin, which was asked by.
Okay.
And the other question that I had was about just in context with Dr. Vishwanathan's question, that is it, I mean, how about using like monoclonal antibodies, which are specific to certain pro-inflammatory cytokines like IL-6 or IL-1 beta? I mean, two drugs are already there on the clinics, like canakinumab, which is IL-1B inhibitor, and tacrolimus or cetralizumab. Oh, sorry, not tacrolimus, tocilizumab and cetralizumab, which are IL-6 receptor inhibitors. I mean, if we go for any specific such pro-inflammatory cytokines. Do you think that will have a better efficacy than steroids, or maybe steroids with them will work in a synergy?
I'm sorry,
Yeah.
I cannot, I don't know. I
Okay.
think glucocorticoid are taken for their anti-inflammatory, but glucocorticoid is more than anti-inflammatory. Glucocorticoids are also very strong metabolic regulator, and the metabolic function of glucocorticoid is not that much understood in the context of Duchenne. So I'm not. So I think anyway, glucocorticoid is what works best and should be, unless we can give gene therapy, but for the meanwhile, it's glucocorticoid. And for anti-cytokine, I'm not qualified to give answer on that.
So I have I mean, the last questions, and one for Sonia and one for both of you. In your myoorganoid models, maybe I missed it, but there's no, I didn't see inflammatory kind of component of your myoorganoid models.
No, not yet. However, I didn't show you other projects ongoing, but we are building a more inflammatory model of organoids, but without adding macrophages, but adding an inflammatory cocktail made up of TNF-alpha and interferon gamma. So with this cue, we see activation of the inflammatory genes. Of course, there are inside always muscle cells and fibroblasts, and also fibrodysgenic progenitors. So we see the increase of inflammatory genes, the classical one, and by adding glucocorticoids as a proof of concept, for example, we show the decrease of those genes. This is because we wanted to have an inflammatory muscle platform to test those drugs. So it is also true that when we boost inflammation, we don't boost very efficiently fibrosis, because fibrosis is regulated more like after inflammation cues, you have fibrosis. So we are setting up a process where we first introduce inflammatory cocktails, and then we introduce fibrotic cue, let's say, to mimic what happened. So for the moment, the idea is to modulate the environment more in terms of extracellular cues, but in the future, we were also thinking to add macrophages to better have a physiological, let's say, triggers of the inflammatory cocktail, of the inflammatory medium. So yes, but adding glucocorticoids in this model, for example, it's enough to reduce those inflammatory genes.
Yeah, thanks. My last question is essentially both to you to start. Your institute, Genethon Institute and Institute of Myology, works on a large number of different myopathies. I mean, like we are interested in
Yes.
GNE myopathy, and your results from LGMD and DMD, and I guess probably there are other myopathies. Do you think there is a common underlying major cause of muscle damage in different myopathies? Or do you think something, is
Yes.
there a common theme running through different myopathies where we can
It's a very
learn
good
from?
question. And I strongly believe that yes. And also, I know that I don't know, I never worked on gene myopathy, but I in contact many years with Israeli researcher Stella, who's working on gene myopathy. And I just noticed a paper that came out on autophagy perturbation in a GNE myopathy.
I know,
And
Stella, we know Stella. So, but you know that,
yes,
no, go
I
ahead,
think
please.
the answer. Yeah, I believe that for many of them. So, okay, let's put it this way. We now screen lysosomal damage over a range of muscle disease, and we see almost all over. This is one thing. The other thing is everybody talk about autophagy, very popular term, but let's extend it into autolysosomal system, which is wider than autophagy, because autophagy is just one aspect of that, so much popular, so much fashion that everybody look on that. But if you took the question bigger than that, then yes, I think it's common mechanism to so many different muscular dystrophies. I'm quite sure that GNE part of them. It's so rare, so I ask people around if they do have anybody GNE transverse section, and no, we had never any, so we couldn't test it. But if we will have any transverse section coming from, I don't think the animal model, to my best of knowledge. But if ever there's a chance to obtain transverse section of GNE myopathy and to test for lysosomal damage, we will at least get answer for this question. But clearly, I think, yes, it's probably the lysosomal system which play important role in this.
I
And
think
in
on
so
this
many
point
other
we'll
muscular dystrophies. I
discuss it
believe,
separately. We can discuss it and take this
yeah,
forward from what you said.
Sure, but yes, there must be a core mechanism in common for so many of them, and many of them, I believe, of the gene, which we don't know why they're causing muscular dystrophies. Some of them will be dealing with maintenance of the autolysosomal system, so there will be defects in the autolysosomal system, and then it may evolve into muscle disease.
Okay, thanks very much, both of you. Ambrish?
Yeah, I'm there.
Okay, so please go ahead.
Yeah, thank you, Dr. David and Dr. Sonia, for such a lovely presentation, which was very enriching and very knowledgeable to us. Thank you for taking out time from a busy schedule and enlightening us with all the research that you have been doing in the organoid models, which we hope will be better translated than the MDX mice models, which are used right now for Duchenne muscular dystrophy. And I also take this opportunity to thank my dear friend, Dr. Professor Derek Goreski, who is not here, for introducing us. And also appreciate Dr. Alok Bhattacharya and Dr. Sudha Bhattacharya for organizing this webinar, which was very, very enriching. Thank you.
Thank you, everyone. And anyway, we hope to see you people soon.
Yeah, it was with pleasure. It will be a pleasure to keep in contact. So
Yeah.
yes, thank you, and see you
Thank
the
you
next
very
occasion.
Thank you. Thanks a lot.
Thank you very much. Thank you.
Thank you.
Bye-bye.